Okay, great. All right. Well, thank you guys for joining and super excited to have Dr. Mahajan here with us today, who is one of our 2024 recipients. And I was we were chatting because she got on a little bit early just so we could make sure all the mechanics of Zoom were working correctly. And I was explaining to her how this award really is so special because there are really kind of people and faces attached to this award. Sometimes when foundations or when there are grants, it's kind of just a large pool of money from very generous donors, but it's not, we won't really know who all those donors are. And in this case, we have our group of family funds who are really, have financially invested and also emotionally invested in this work. So it really does make this award really special. And thank you guys all for adding your because ofs. To again, just kind of remind us why we're all here. And I did want to just take a brief moment to introduce Dr. Mahajan before she goes into her overview. But really excited to have her here. And she is going to be joining us at Factor for the live in-person check presentation. So excited for her to soak up kind of everything Factor has to offer. She is a professor and chair of medical laboratory sciences at Hunter College at the City University of New York. And she has a MS in biochemistry from the University of Hyderabad and a PhD in molecular biology from the Indian Institute of Science in Bangalore. And she's completed her postdoctoral fellowships in molecular biology at both Weill Cornell Medical College in New York and NYU Medical Center. And her lab's current research focus is investigating drugs that are effective in killing metastatic osteosarcoma cells. And the lab is currently working to determine the mechanism of action for riluzole in osteosarcoma. And so I think that's what she'll be kind of giving us an overview of today. I also just wanted to note that she mentors undergraduate students and PhD students in her lab. Her students are from a diverse population and among minorities, especially women in the sciences. And currently she has 3 PhD students pursuing their thesis research in her lab. So it's also wonderful to have someone who is a dedicated mentor and dedicated to kind of just creating that strong pipeline of researchers in this disease. So thank you. So with that, Dr. Mahajan, if you want to go ahead and you can start with your overview. And guys, I mean, we're such a small group live that feel free to ask questions. Is that okay, Dr. Mahajan, along the way?
Absolutely. Thank you so much, Christina, for this beautiful introduction. And I'm truly deeply honored to be here and to be, selected for this award. It's a huge blessing, and I share your grief, and I really truly am very thankful for this award and this opportunity to present my work. And please interrupt me with any question. It's not interruption. You can stop me with any questions, and I'd be happy to answer more elaborately at the end of the talk. As Christina said, I have a faculty appointment at the professorial level at Hunter College in New York. And sorry, I'm just getting my laser pointer. And I have an appointment at the graduate center. That's how I have doctoral students in my lab doing their PhDs. These are, so the department is in the downtown area of Manhattan. The graduate center is in the Midtown, and my lab is actually uptown. So it's a bit of a commute pretty much most of the days, but I'm very happy to have this position and be able to do this work. I'll start off with a little bit of like a history. So you see here, this is Lou Gehrig, the baseball player who played for Yankees. And at the peak of his career, at the age of 36, he was diagnosed with ALS, which is the amyotrophic lateral sclerosis. And that disease now is popularly called as Lou Gehrig's disease after him. And he gave a speech at the time of his diagnosis when he was forced to retire. And he said he was the luckiest man alive. And a year later, he passed away. So ALS is a progressive neurodegenerative disease. And one of the symptoms, or rather one of the indicators, is that the cerebrospinal fluid has elevated glutamate levels. So almost about 5 and plus decades later, a drug called riluzole, which is a benzothiazole, was tested in a clinical trial in 1994 and was approved by the FDA as a drug to treat ALS. It's also used, it's used currently for ALS, and it's been tested for several psychiatric disorders. What riluzole does is block glutamate release and basically bring down the levels in the CSF patients. Because this is a progressive disease, the patients generally live between 3 to 5 years post-diagnosis. However, it actually delays tracheostomy in these patients. So, it's helpful nevertheless. So riluzole not only blocks glutamate release, but it also actually helps clear the glutamate from the junctions where it activates the cells. Riluzole is available as tablets, a 15 milligram dosage, which is given twice to ALS patients. It's available as a liquid form for patients, who can't swallow tablets, and also available. It was introduced in 2021 as an oral film for patients who can't swallow because ALS it's a neurodegenerative disorder that actually kills motor neurons. So basically, the muscles become very weak, and the patients can't swallow even at some stage in their during the disease. So Sir James Black, who was a Nobel Prize winner in 1988, he said that the best way to discover a drug was to start with a new one. And what we're doing in our lab is actually to repurpose an old drug. This also has fiscal bearings because when you have a new drug, it costs from start to finish of the, for funding of all these drug investigation from the lab onto clinical trials and in the clinic, it takes about 1.4 to 3. Sorry, I just want to change my display here. So I can deal with this. Sorry, so going back to the fiscal benefits is that for a new drug, it costs about 1.4 to 5 billion dollars, as opposed to a repurposed drug, which only can be between 52 to 81 billion. And this is according to a study that was published in JAMA Health Forum. And this is all the money that was funded by NIH. So for the survival of patients with osteosarcoma, we all know this. This is just to reiterate that we need new drugs because the 5-year survival rate hasn't improved since not significantly improved since the 1980s. So we are looking for better drugs to do better than what we're doing right now. What is the rationale for repurposing riluzole? Well, there is a decades of success in various neurodegenerative diseases, and we know that it has low toxicity and good tolerance. And it works better. It works really well with cancer drugs, which we know from some of the clinical trials that have been conducted, and I'm going to share some of those. It has high tolerability in cancer patients, especially with melanoma and glioma, has been tested more than in other cancers. And it also was shown to be a radiosensitizer in nasopharyngeal carcinoma. And we do have some results about that I'm going to share in a few minutes. So what is the connection between glutamate and cancer? So glutamate signaling is abnormally activated in cancer cells. There have been many different types of cancers that have been studied, which actually release glutamate into the extracellular space that is outside of the cells. And then the cells use that to signal their growth via these receptors. All these receptors have been identified and studied in various different cancers, and basically leading to cell proliferation, migration, and angiogenesis, which are basically the hallmarks of cancer. So what riluzole does is block the glutamate, thereby blocking the signaling and thereby blocking cell migration. But what we also, cell migration, cell proliferation, and angiogenesis, but we also see that when it does that, it induces apoptosis in most of the different types of cancers. Here is a list of all the different types of cancers. There are 11 listed here. I'm not going to read the list. And these are the publications that basically have shown work with riluzole. And I'm going to show you a snapshot diagram that basically details some of the work. And my lab is the only lab that is actually working on osteosarcoma with riluzole. And we have published a few papers since we began working on osteosarcoma in about, I think, mid-2014 or 2015, early 2015. So summarizing the work from the testing of riluzole on different cancers, we had actually published this review article in 2021 during the pandemic, and this is a beautiful rendering done by my graduate student. I don't want to go into details, but it shows is riluzole basically targets very different pathways in the cells, and this is basically like a snapshot of what happened in different type of cancers. All this doesn't happen in one cell, but in various different types of cancer cells. So what we found in osteosarcoma is that the glutamate receptors are activated. And when you block that with riluzole, it induces apoptosis or programmed cell death by activating phosphorylation of this protein called YAP. And the BAX expression, which is a protein that executes apoptosis, is activated. So this was the finding from our lab that was published in 2021 as well. So just to go over some of the key features that riluzole acts on is that it blocks signaling, it blocks calcium increase inside the cell, it increases oxidative levels, basically leading to DNA damage in the cell, causing cell cycle arrest, which means that cells will not be able to divide anymore. And more recently, we found that it suppresses the activity of an enzyme called matrix metalloprotease 2, MMP2, and I'm going to show you some data about that in one slide a little later. And I just told you about the S-associated protein, or the YAP, that is phosphorylated to induce apoptosis in osteosarcoma. So I just want to show you some of the clinical trials. I just want to tell you all that I tend to speak very fast. So if it's too fast, you can stop me. Is that okay? Or are you able to follow me? Perfect. Thank you so much. Appreciate that. So I want to show some of the clinical trials that were done in other types of cancers. For example, in melanoma. Melanoma is one cancer in which a lot of work has been done by riluzole by Susie Chen's group at Rutgers, and I've been following her work since I started working with riluzole and osteosarcoma. And basically, why does riluzole work in melanoma? It's because 60 to 70% of melanoma tumors actually secrete glutamate, and that's why this drug is very useful. They had done the phase 2 clinical trial in 2018, and what they found was when riluzole was administered at 100 milligrams per day for 14 days, it suppressed the tumor growth and also blocked the signaling, which is known to be activated in vitro culture, which means in the laboratory. So what they had found in the lab was also true in human patient samples, the patient samples. So that was a good finding. More recently, there have been phase 1 clinical trials with riluzole in combination with sorafenib, which is basically a kinase inhibitor in patients with solid tumors. So this study had a lot of different type of cancers that they had studied, that they had included in the study. They used riluzole at 100 milligrams per day, and they used sorafenib at 400 milligrams in the morning and 200 milligrams in the evening. The study caught my attention not because it had riluzole, but because they had 2 cases out of 35 cases that were sarcoma. They didn't give details of what the sarcoma was. It could have been any sarcoma. And the good news is that those 2 patient samples showed increase in markers for apoptosis, which is cell death. So this is our work with the osteosarcoma, which we started, and this work is almost close to being published. So we actually have a manuscript ready to go any day this week or next week, early next week. So what we've done is we picked cell lines, pair of cell lines, non-metastatic with metastasis. So we have 1, 2, 3 pairs. So these are the parental cells that are non-metastatic and artificially made into metastatic by injecting in mice and isolating those cells. And we also took non-tumorigenic cells, which are just immortalized, which are the fibroblast, fetal osteoblast. And then we have U2OS cells. These cells are a little different, although they're used for osteosarcoma studies, that they have little different features than osteosarcoma cells. And then we use 2 mouse cell lines just to make sure that what we are doing in human cell lines basically is true for across species as well. Then we included 4 different PDX samples, patient-derived samples, of which one of them is a metastatic one, and these are the mutations that are seen in there. I'm not going to go into details. So those are the cell lines that we tested. Because we were going to test with riluzole, we had to test if these cells actually secrete glutamate, and it turns out that they actually secrete glutamate over a period of 7 days. in all of those. So these are the primary tumor cell lines, the metastatic cell lines, and here are the PDX cell lines. A couple of the PDX cell lines don't secrete glutamate as much. They're on the baseline, but 2 of them do. So, I did tell you that the signaling happens in cancer cells via glutamate, receptor activation. But how does the glutamate receptor actually activate the pathway? So what it does is, here is one of the kind of the glutamate receptors, which is called the metabotropic glutamate receptor, and there are many different kinds. And I showed you a slide with different isotypes were expressed. So what this does, many of these different functions here, the ones that are involved in making the cells divide and proliferate to make the tumors grow, is the sustained proliferative signaling. They resist cell death, so they just continue to exist and survive and thrive. And then they also, the DNA gets replicated, so it helps with the proliferation or growing of the cells, dividing of the cells, and they evade growth suppressors. So normally cells would have something that will, the molecule that basically will block growth, and the cancer cells are smart and they find methods to actually block those proteins so they can continue to divide. So those are the 4 things that are targeted if you use riluzole on the cells. And when we did an assay to look at growth inhibition with riluzole, we found, so these are all this 15 cell lines that we have, 11 cell lines and 4 PDXs. All of them show growth inhibition. So with increase in dose, so this the starting lowest dose to the highest dose, they show over 80% of cell death or other inhibition of growth. One of the properties of cancer cells, common knowledge, is that cancer cells basically leave the tissue where they're growing. This is the tumor, primary tumor, and then they break the tissue barrier, get into the bloodstream by intravasation, and then they get out of the bloodstream by extravasation, and then they get lodged into the site of preference for metastasis. So basically, cell migration and invasion are 2 properties of cancer cells that are crucial in setting up the metastatic process. So we wanted to study if riluzole had any effect on that. And so glutamate receptors are also known to activate invasion and metastasis. So we wanted to test the effect of riluzole. And we did this assay called invasion assay. You do this assay in basically in the dish. What we found is that this is just a control where cells are not expected to grow. This is a normal cell without any drug, and the green bars are the ones where riluzole is been treated in the samples. So you see all of these cell lines. So these are different cell lines that I showed, the 4 of the 11 ones that I showed you, they all show reduction in the invasive ability. So this is good news because this tells us that riluzole actually blocks the, glutamate. It may be blocking through glutamate receptor. We haven't tested that, but however, it does block the invasive ability of these cells. So is true for the PDXs. These are the 4 PDX cell lines that we have here. They all are inhibited by, they all actually are reduced by glutamate treatment. Sorry, riluzole treatment. And so our question was, if riluzole is blocking invasion or rather reducing invasion, how does it do it? There are 2 proteins in the matrix, which is, so you have cells and you have the matrix outside of the cells, which is made up of proteins again. And one of the main protein in the extracellular matrix is collagen. So collagen actually keeps the cells in place where they are. So there are 2 proteins, the matrix metalloprotease MMP2 and MMP9. There are 2 collagenases. That means they break down the collagen, which facilitates the cells to actually move out of the local primary tumor site and then move outside of that region. So we tested MMP9 and 2. 9 was not affected. However, MMP2 in most of the cell lines was reduced when we treated with riluzole, but not in LM7 cells. So it turns out that in most of the cell lines, riluzole acts by reducing the activity of MMP2, which is the collagenase activity. So that's again good news because we know in some ways how it prevents invasion. So what I showed you so far is in vitro data done in dishes in the lab. We also tested riluzole in animal model. So this model basically is a dirty model because we actually inject the tumor cells in the tail vein, so it gets into the bloodstream, and then, of course, it goes through the heart and lodges into the lungs. But the cells will start to preferentially grow in the lungs. As we know, it's the preferred metastatic site. So once on the day 3 after injecting these cells, we started to so these cells actually have an enzyme that makes the cells give out light, so you can capture these images in a machine to see where the tumors are growing. So when we saw that the mice that had the signal, we collected them and then randomly divided them into 2 groups. One group did not get any treatment; they just got just the PBS, the vehicle, and then the second group got riluzole. And we did see a significant decrease in the metastatic tumors in this model. So what we did further was to take the tissue, lung tissue, and make sections and stain with this stain for this protein called nuclear mitotic apparatus protein. This is a great indicator of how many cells are dividing in the tumor tissue. And we see that this is the control sample, and this is the one that's treated with riluzole. We see that the number of positive cells decreased significantly, showing that riluzole is actually able to decrease the number of dividing cells. So this is, again, a very good data because it basically supports what we found in the dishes. Now, there is a little bit of a problem with the riluzole in the sense that when riluzole is given to human patients, they show differences in the exposure to riluzole because there is this whole big superfamily of enzymes called cytochrome P450, which are basically involved in metabolizing drugs. That means they actually break down the drug to clear from the body. And one of the enzymes, which is a liver enzyme, is called CYP1A2, and this directly metabolizes, or rather breaks down riluzole to make it into a compound that can be eliminated through urine. Now, why is this a problem? Because this enzyme shows heterogeneity in human population, which means some patients, some subjects have higher levels of CYP1A2, and some have lower, because of which riluzole gets metabolized differentially, thereby the effective dosage, or rather dosage is same, but the effective exposure is different. Therefore, a group by Alan Reitz basically made prodrugs of riluzole, which is a dipeptide prodrug. One of them is called FC3623, and the other one is riluzole. And these drugs, because they're dipeptides, they are, they're given orally, of course. So they go into the stomach, right? Once the digestion occurs, it's absorbed by the portal system that takes it to the liver. And these two peptide drugs are not, they're resistant to the degradation by CYP1A2 enzyme. Thereby, they go as dipeptides into the bloodstream and to the cells, where the cells basically cleave the dipeptide and release riluzole. So this actually, once the oral dosage happens, it takes about 2 hours. In clinical trials, that's seen that it takes 2 hours for the riluzole to be released from this, thereby enhancing the availability and bypassing the hepatic clearance of these drugs. So here is the study by Jeff Pelletier and Alan Wright's group, where they actually took they tested riluzole and prodrug FC3423 in mice, where they injected the mice with the tumor cells, and they followed up over 21 days. And you see the blue bar is without any treatment. Whether you use riluzole or FC3423 compound at 2 different doses, you see that the tumors are reduced to 10% of what grows in control. So basically, this drug is as effective, or that dipeptide prodrug is as effective as riluzole. So I want to make a point here that in animals, in mice, this enzyme doesn't exist, so it doesn't matter really, but in humans, it does matter. And here is the second drug that I mentioned that this group had synthesized, which is called troriluzole. And this group, this group actually did the clinical trial with this. What this clinical trial was basically to see what is the maximum tolerable dose of troriluzole. And they found that they could use up to, they actually used up to 420 milligrams in the highest cohort of the drug dosage in patients. So that was the dosage. And that dosage is about 8 times higher than the dose of ALS, what's given to ALS patients. So, and like I said earlier, troriluzole was converted to riluzole in 2 hours. So is FC3423 compound. Now, I just want to highlight the aims that I have proposed in the award for the proposal. We actually aim, we have screened 4 PDXs, and I've shown you the data. What I plan to do is screen 4 more at least, if you have more. And in fact, the study beyond the award would be to screen more drugs, and I will get to that point. So we want to screen 4 PDXs. And we want to not just screen with riluzole, but with prodrugs also, so that we can establish the dosage and the success of prodrugs with these PDXs. And these will be new PDXs besides the ones that we have in the lab. We will be testing this in vitro in the lab in the dish. And then the second part of the aim 1 is basically to create a table like this. So these are the PDXs that we already worked on. And here are the metastatic. So this is the only one that was metastatic to begin with, was isolated from lung. However, OS69 is a PDX that was isolated from a primary site, but in animal model, it goes to both lung and liver. So it's a great model for us to study and prevent and see if riluzole prevents the metastatic process. So that's something we want to do. And of course, in vitro, they're all inhibited by riluzole pretty significantly. So we're very excited about that, and we want to make sure that we actually riluzole is efficacious for more PDXs as we test them. So for aim 1B, we would like to increase, keep adding to this table and have a table with the profile, mutational profile of the patient, whether or not they are metastatic in nature to begin with, and then to see if they are efficacious, if riluzole is is inhibiting the growth in these cells. Aim 1C, we want to basically try this, once we screen them in the lab, in the dish, we want to see if we can see the efficacy in vivo in animals. So we will at least choose out of the 8 PDXs that we would have, we will choose 4 PDX lines against FC430, against riluzole and FC3423. We will analyze the tumor burden along with the tissue analysis with several markers to see if the tumor cells are being reduced, if they are undergoing apoptosis, etc. So I would like to address some of the weaknesses that were pointed out by the reviewers, and I'm very thankful to them for that. And I was kind of aware of that, but here are my answers to those. I've just put on their comments as is verbatim, and I just highlighted what they said. So the question is, if the osteosarcoma is known to be, not have any druggable targets. So what are you going to do with this kind of a problem with limited single agent efficacy? So what I want to point out is that when I showed you this data, I didn't highlight one of the cell lines. So all of these cell lines actually show, this one has a high IC50 value, but there's one typical cell line which shows a typical behavior, which is 143B. This was a derived cell line from the HOS cell line. And it showed, even at the highest dosage of riluzole, it showed only 50% cell death, or rather inhibition. So we were curious to see, like, this was the only unique one that behaved that way. So we, of course, looked into it in more detail. And we know from the published literature that this particular cell line carries a KRAS mutation. And we were also aware that in melanoma, a mutation in KRAS, BRAF, or PTEN actually conferred resistance to riluzole, which means that they were not as effectively killed by riluzole. So then, so because of the IC50 value for riluzole very higher, we thought we should target this KRAS G12S mutation by using a very specific inhibitor. And when I presented this work at college in one of the departments, one of my colleagues actually, it caught his attention because he works on KRAS-driven mutation cancers that are driven by KRAS mutations. And he told me about a drug that was just published. And that drug was like a new generation drug and was very specific to this particular mutation. So we did this work in collaboration with him. The drug is called G12SI5. And we basically tested combination of the drug with riluzole. These are the increasing concentrations, just showing the scheme of how we did that. And what we found was that this is the parental cell line where the combination of drugs doesn't do much because these cells are killed by the single treatment of single agent, which is riluzole. Here is riluzole. This is how G12SI looks. And to our surprise, these two drugs together in the 143B cells show synergy, which means the effect of individual. So the effect was more than the individual drugs as well as the additive. So basically, when the effect is more than additive, you call it synergistic. So we got synergistic effect. And in fact, this is the zip energy scores are showing in this graph. The host cell line, which is the parental cell line, doesn't have a synergy score that is good because they don't synergize the drug in killing the cells because the cells die with single agent. However, the zip score for the combination drug for the 143B is 1.8, which is good because they are better killers together for the cells. So, as I told you, this cell line was created from the parental cell line. So what is the relevance to osteosarcoma patient population? This is an old study that was published in 1998, where they had actually analyzed 17 osteosarcoma patient samples. They did a genetic analysis, and they had found KRAS mutations exactly at the same location. I think it was G12S and G13. So 2 out of 17 patients had the KRAS mutation. So I believe that there are more out there. It's just that we haven't looked closely enough. So this is the relevance that if patients have KRAS mutation and they are not effectively killed by riluzole, then they can be specifically targeted with the drug combination. So the other concern was how resistance mechanisms will be predicted and what they intend to do with this information. So I showed you this table already, and I said that only if riluzole is not killing these cells effectively, then we will look into the mutational profile more carefully and try to find any specific mutation that could be targeted with a specific drug. And we would hopefully, I have some candidates in mind, I just don't have them on the slide, that I would want to try FDA-approved drugs just to speed up the process from bench to bedside. And of course, we will create this table and we will use the information to find specific targets. Although the whole idea of starting this project was to see if riluzole can kill all osteosarcoma cells with different mutations because of the heterogeneity, and it seems to be true. So out of 15 samples that we have screened, there's only one that shows a high IC50 value. So this was more of a suggestion, is that we need to screen more PDXs in order to validate the efficacy of riluzole, before we go ahead with the clinical trial. And of course, absolutely, the more, the better. And if we do this, then we have done our homework well before we go into a clinical trial, hopefully sometime soon. And the other question was whether we're going to be using multiple agents together in combinations. I would want to start off with 2 agents at a time in combination, and then we'll evaluate. And then if necessary, then we could add in more agents. But to begin with, we want to do just two drugs. So it will be riluzole plus another drug. So besides the work that I presented, which formed the basis or the rationale for the proposed work, we have some exciting findings in the lab, which is that when we treat osteosarcoma with riluzole, it increases this protein called EGR1. Now, why is this important? Because there was a paper that published that any of the so there were like 2 or 3 cell lines that they tested. It was not done in patients. What they found is when they treat the cells with any of the chemotherapeutic agents such as methotrexate or doxorubicin or cisplatin, they actually saw the EGR protein go up. They didn't look at mRNA. As far as I can remember. So anyway, the protein expression was increased. So we had actually, based on that, we wanted to see if riluzole can induce EGR1 expression. And EGR1 is involved in execution of apoptosis in osteosarcoma. We have shown that. And also EGR1 is induced by what is called as reactive oxygen species, which is one of the first markers that is elevated in osteosarcoma cells when we treat the cells with riluzole as a matter of 30 minutes to an hour. So what we are proposing is to use this as not just a diagnostic marker, but as a treatment marker, as a therapy marker, and we are working on that. So we have done work in vitro, and we have some exciting in vivo data. The other exciting project that's going on in the lab is that I mentioned early on in the slides that riluzole was shown to be a radiosensitizer for nasopharyngeal carcinoma. And based on that, we wanted to test. And what we have found is we have very exciting results that riluzole actually radiosensitizes human and mouse osteosarcoma cells, which are metastatic to radiation. So radiosensitizes, of course, to radiation. So that's very exciting. And we haven't done in vivo work yet, but we are pretty close to doing that. And part of that project is actually looking at how does riluzole radiosensitize the tumor cells to radiation. Sorry, radiosensitize. What we found is that it actually does it through VEGF-A, so it brings down the VEGF-A levels, and it also seemed to induce markers for apoptosis. So this is ongoing work, but this is very, very exciting data that I couldn't help but share with you all. What are my future goals? This is just a big dream, and you can't do anything without having big dreams to begin with. So sometime in the near future, clinical trial with either riluzole as a single, or maybe that's just too much of a positive thinking, but I would say in combination in osteosarcoma patients. Maybe someday use glutamate signature as a to basically have personalized medicine, where if the cells secrete glutamate, can be used, riluzole can be used for these patients. Of course, when there is a glutamate receptor activation, etc. So those can be tested easily, and then that's something that we hope to do. I'd like to thank my collaborators, Dr. Healy and Raj, a great team. Dr. Healy's lab has been a big supporter, and he's the co-principal investigator, and his lab is the one that's providing PDXs, and Raj is the one who actually isolates the cells from the patients and brings it to my lab. So I'm super grateful to their team, to Dr. Healy, for having trusted me with his precious samples, and I'm very grateful for that. And Andy Wolf is my colleague who actually told me about the KRAS inhibitor, and we did that work together. And we actually have a side project going on that we will be publishing soon, that actually delineates the mechanism of how this works. These are the people who have worked directly to generate this data. Oken and Mariam are two of my graduate students. They've done most of the work, and Shraddha has done the glutamate assay and the MTT assays. Beatrice has done one of the assays. She's an undergrad. Shlomo Pelas is an undergrad who actually did the KRAS inhibitor work along with Mariam. So I'm very thankful to this team. Besides these students, I'm not going to go through the list. Here are the students who are in my lab. Pooja is another graduate student, and the other students are either undergrads or MDs. And these two students that I listed in the end are current high school students. I'm happy to just get them all excited about this research. And thank you all for your attention.
Thank you so much, Dr. Mahajan. I have to say, I'm feeling super pumped after hearing your presentation. And this time is really for you guys. If you guys have any questions for Dr. Mahajan, feel free. And we're a small group, so if you can just unmute.
Yeah, as many questions I can answer. I'd be happy to.
So I have a question about the availability of the prodrug and the regulatory process to get that out. I assume it's not approved right now. Is it available?
Yeah, that's a great question. So I'm in contact with Jeff Pelletier, the first author on that paper, and he actually supplied the drug to my lab, free of charge. And he was also interested in doing, depending on our results that we get, interested in doing like a business grant with the NIH. So that's something that I need to explore more once the once we test it and we have good results. So they are actually synthesizing this from this Biohaven Labs. So Alan Reitz is the one who owns the company. So yeah, it's not available in the market.
It's not available in the market, and would it be available for human use in a short time, or is it like, I mean, what's the process to get it into humans?
I won't be able to answer that, but since they're doing clinical trials, I'm sure they are working on that process for human use. They have done clinical trials with troriluzole, I know that, and they were in fact running another trial sometime during pandemic, and they said that they had to drop, they had to discontinue the trial because of low enrollment. So I know that I really don't know the answer to that, like when it would be available, but something that we can work on with them if the results look good.
So I have one other question. So part of Paxlovid is a CYP inhibitor. So is it possible that you could look at CYP inhibitors as well as in a combination kind of therapy?
Absolutely, yeah, we are open to trying. So one of the things I wanted to do was just screen like a lot of different drugs against different kinases and like just pick battery of different types of targets and do the screening. But it didn't seem like a well thought through plan, because we're just doing exploration. But certainly we can try inhibitors in combination, and it's easy to test it, so we'd be happy to.
Sounds like a good idea. Paxlovid, I think, is a repurposed HIV drug from Pfizer.
Yes.
So
Yeah.
I think the concept of repurposing these drugs and using them in kind of unique combinations is a really great idea.
Thank you. Thank you. Yeah. Okay.
Biohaven is actually a pharmaceutical company.
Yeah.
I'm going on their website, so I'm trying to dig
sure.
a little bit further. So they're a commercial and research-based organization. So they likely are. I'll have to dig into that and see. I might be able to get us some more information on that as well.
That'd be wonderful. So I've spoken to Jeff Pelletier a couple of times on Zoom, never met him. And he worked with Alan Wright. So they have done clinical trials with Susie Chen's lab. So I know that they basically use research from research labs from universities to test their drugs. So they are really into it. I mean, whatever help you can get us through your network would be wonderful.
So the presentation that you did, and thank you very much. This is Rob and Karen Marsh, by the way.
Nice
thank you very much for presenting your information. And much like Christine, the information was very intriguing and very positive in what I could see. You'd mentioned a couple of sidebars with regard to this KRAS inhibitor, the one that your colleague had, I guess, introduced to you. So and I apologize, I'm not looking at the paperwork, so I'm curious, is that KRAS inhibitor that you just referenced, is that going to be part of this study that you're doing? Because you've referenced a couple of different, maybe I just didn't focus long enough, but it sounded like you had a couple of different things going on.
Yeah, so the study is going to, I just showed that as an example. It was part of the preliminary data for the proposal, so that's why I showed it. To also make a point to the comment about the weakness in the grant is, what are you going to do with the information if you knew there was a mutation? So I wanted to say I could use that information to specifically target a certain mutation, and that was a good example that we had already tried. But it's not going to be part of the study because, well, of course, if the PDX has that mutation, we will go ahead and test it against that. But if not, then we won't. Yeah.
Okay. Thank you.
You're welcome.
Any other questions? I might throw out a couple, but feel free to cut me off if you have a question. You had mentioned, Dr. Mahajan, about like 60 to 70% melanoma secretes glutamate, and I might have missed this too, but did you mention what that is for osteosarcoma?
From our study, from the cell line that I showed you, I'm actually going to show you that. Oops, I don't know how to. Okay, I'm just going to. Oh, okay. I'll go back to the slide where I showed the
Okay, great.
glutamate secretion. Yeah, just so that I can, make a point, basically. Where's that? Right there. So here's the, all of the cell lines secrete glutamate, and they all, you can see that they are varying degree. These are primary and these are metastatic cell lines, and here are the PD axis. So of the 15 different cell lines that if we call them all cell lines, 13 of them actually secrete glutamate levels that are significantly higher. So yeah, and this is there was no such data earlier, so we are the only ones who've seen this. So we will continue to see if measure glutamate levels in PDXs as we go on, because it then makes. But the other point that I need to make is that riluzole seem to actually not only block glutamate signaling, but also induce like a reactive oxygen species and oxidative stress in the cells by blocking another transporter, which is common in most of the cells, but seem to be overexpressed in cells, and especially in osteosarcoma. And we showed that we have the data, but I didn't show you here. It's called the XCT. So the short answer to your question would be yes, osteosarcoma in our lab have shown glutamate secretion. Pretty much most of them except the 2 PDXs.
She mentioned something, so I will ask her. I think she did mention. Was it okay?
Hey, Rob and Karen, just so you know, we can.
Sorry,
That's
Rob's
okay.
looking at, he's looking at something, and I'm like, I think she already
no
mentioned it,
worries.
but we can't see the slide, so I apologize.
No, don't worry about it. Anyone have any other questions? All right, just throw another one out. So I was just curious because, well, so first of all, this is so exciting because of the translational potential, just because of the repurposing, and I thought it was really interesting that you threw in kind of the financial implications there, but also just all the learning that you're able to build on from if this drug being studied in other diseases. And so you had talked about the EGR1 and how MAP kind of have is kind of high because that it's increased by chemo. And I'm wondering, when you think about translating to this clinic, kind of where do you put this in the treatment like sequence? Is it a maintenance drug? Is it something that might have synergy with MAP given the EGR1 thing? Like where do you think, like as you plan towards a clinical trial, kind of where does this go in the treatment?
So that's a great question. So if let's say we are doing a clinical trial with riluzole, and I think that single agent, we all know, no secret, the single agent efficacy is not, something that we can trust. So most likely we'll come up with a combination, or let's say even if it's just riluzole, we would like to use EGR as a marker to basically study, like it's an early marker in the apoptosis process. Because there will be other markers that are well known, in research that can be used as apoptotic markers, but EGR is specifically induced with this. So we could use so you could take biopsy samples after treatment and check the EGR in levels in the samples to see how well the patient is responding to riluzole. I mean, that's our hope.
And then because you kind of reference this a few times, because it does induce apoptosis, but it sounds like it's more effective at inhibiting growth and inhibiting.
Oh yeah, so yeah, that's a great question. So in our previous study, when we had just looked at 2 cell lines, one mouse and one human, we did apoptotic assays to screen to study efficacy of riluzole in those cells. But those are very, very tedious assays. And that's why we reverted to this. We actually moved to this assay called MTT assay, which we're looking at growth inhibition rather than apoptosis. But we very well know that riluzole induces apoptosis. So when I say growth inhibition, although it should not be, but for our purposes, it's equivalent to inducing apoptosis. Yeah, it's not technically correct to say that, but it's equivalent to that, I can say, from our experience, because we have done this a million times.
Any other questions? Can I just ask one more in honor of my dog's birthday?
Please. Please.
Is what about, because you talked about, one of the steps is animal models and mouse models, but I'm just curious, is this something that could be applied to canines as kind of a step to get you to human trial? Would this
Yes.
be active at all in canines?
Yeah, absolutely. So, I don't know the answer, but because I haven't tested with the canine, but we have a mouse that's like far off, and then the dog is in between, and we are like far ahead, right? So I'm assuming, or rather my wishful thinking is that it would be effective, but we have to try it out. And I think the clinical trial with dogs from a vet will be a great one to try out, because I think that dogs are more prone to osteosarcoma, so that'll be very helpful. So that's something that actually I haven't worked on yet, but Dr. Healy and I discussed about it, and he was the one who proposed that we should look at it. So I said, that's a great idea. So I know that there's a grant out there that I should have applied to, but I think the deadline is still, not passed, so we could still do it. We'll see. Yeah. It's a small grant, so I was thinking, do I have time to do this, etc. But it's something to pursue seriously. Yeah, thank you so much for supporting that.
Yeah, I was gonna say there'll be a bunch of vet onset factors, so it'll be a good place for you to maybe shop that idea around and see.
That would be wonderful. Yes, absolutely.
All right, guys, any other questions? All right.
Not at this
I
time.
don't think-
Thank you, though.
All right.
Thank you.
Just
You did a really nice job.
It was a great presentation.
Thank you. Appreciate it. I really, really do. Very, very thankful for all your contributions and everything that you do. And I was telling Christina that I really admire her for doing all this work that she does. Amazing. Thank you all so much.
Thank you. We appreciate your work.
Yeah, very good.
As well as the
Thank you.
presentations.
It'll all be worthwhile when it goes into treatment, one day. Yeah.
Rooting for it.
Yes, thank you.
Okay, thank you guys all for joining on a weeknight evening, which I know is difficult. Really appreciate it. All right, thank you.
Thanks,
Bye.